Falling Walls Circle, Challenges and Foresight.
Welcome to the Falling Walls Plenary Table, the future of imaging in the age of AI.
Hi, I want to welcome all of you for coming here today. My name is George Caputa. I am a senior editor at Nature, and I handle papers involving applied and clinical AI. We have a really incredible panel here for you today to discuss the future of imaging. We have Stephanie Adi from BioHub, where she serves as the vice president for imaging science. We have Martin Hetzer, the president of the Institute of Science and Technology, Austria. Jason Sweeto from BioHub, where he serves as the senior director for bioimage data. And we have Paul Matthews from the Imperial College London, where he serves as the director of the Rosalind Franklin Institute and is a professor of translational neuroscience and therapeutics in the Department of Brain Sciences. And so today we're going to ask a couple of questions, talking about the future of imaging and probably quite focused on AI. And I'm going to ask all of you some questions, and feel free to chime in. Let's get some discussion going, and then we'll take in some questions from the audience. So I'm going to start with you, Stephanie. Why is imaging and image data so important to scientific and clinical research? And this is probably something that each of you can answer. Can you give your favorite example currently of how AI has accelerated discovery in this space?
Yeah, so at BioHub, what we're focused on in our broader mission is to cure, prevent, or manage all disease. And our ability to do that will depend upon us understanding complex systems at a much better level than what we do right now. And imaging allows us to have these windows into the complexity of biology. We can start to view cells, the inner workings of cells, the inner workings of the body across both space and time. That gives us an understanding of how the systems are operating and where to be able to intervene for optimal functioning.
Yeah, and do you have an example of just how in the recent years, you've just seen something incredible, a leap and bound in terms of our ability to image or what we can image or how we image?
Yeah, so we have been investing in a number of different technologies, and I'll give examples kind of on the two ends of the spectrum. So we've been investing in cryo-electron tomography, which is a technique that allows us to visualize the molecular motors inside the cell. And this is, right now, just a few years ago, we could only image a fraction of the proteome, a fraction of these molecular motors. And we're creating technologies that will open up this entire inner workings in the cell. And AI, now applied to these questions, is dramatically accelerating our pace of what we can visualize and what we can start to understand. And then on the opposite end of the spectrum, there's new technologies that we're developing to be able to visualize cells and their signaling processes deep within the body. So before, you would only get these high resolution or low resolution images of the body, and now you can start to visualize on a cellular level.
So we're really able to construct detailed maps of what's going on within a cell, and then within a tissue, within an organ.
Yes, and within whole organisms.
Within a resolution that we really were unable to do before.
Correct.
Yeah, it's quite interesting. How about any of you? Is there just something that there's been an advance in the past 2 to 5 years that has really just kind of wowed you?
Absolutely.
Yeah.
Well, one for me is also coming from neuroscience. Excuse me. When one of the biggest challenges still is to map the synaptic connections in the human brain. Still challenging in the mouse, but in the human it's even more challenging with trillions and trillions of connections. And that has been the field of electron microscopy, where painstakingly cut sections, low volumetric analysis of the brain. And that now with a whole set of combinations of new methods, such as expansion microscopy, transferring this to light microscopy, in fluorescence microscopy. This can be now with AI accelerated to a pace where it's actually now for the first time really feasible to even map the entire human brain.
So at this point, we're talking about like layering on different imaging modalities.
Well, this has always been, I mean, light microscopy has been around for hundreds of years by now, and we keep layering on and also inventing new modalities. And I think, yeah.
Go ahead. I mean, I think one of the other really important things is that AI is now transforming the way we image. So for one thing, we're reaching physical limits in many of the tools that we use for imaging. And what AI is helping us do is transcend those physical limits by integrating the prior knowledge that we have from all of the other images that we conducted before. It's just like what happens when a radiologist comes to an image clinically. You don't look at that image as if it was the first time you'd ever seen it. You carry with your, the radiologist carries into that image all of the experience of the prior images, and those are used to enhance the resolution and meaningfulness of the individual image. So we're seeing that in clinical imaging. We're seeing it down at the level of cells in cryo-electron tomography and imaging. You can see things that you've never seen before by applying the AI as ways of enhancing information content.
And to be specific, George, I think what's happened, in the last 5 years. If you look around this room, if we had an image of everything and everyone in this room, and we wanted to identify everything automatically, we actually realized that AI can do that with most sort of conventional images. In the last 5 years, that started happening with images of cells and the volume data. So with pretty good accuracy, not perfect, but pretty good accuracy, better than 95%, we can identify most of the objects in these images of cells and tissues very, very quickly.
Nice.
Right? And then, as Paul says, then there's the things there's the novel things that we don't know that we can identify. That, the speed at which we can work has vastly accelerated.
I mean, a lot of the capabilities of these models, right, is in how they are built and how they are constructed. And the foundation of any good model is good training data. And so we published something last year saying garbage in, garbage out is kind of was if you see the cover, it was kind of funny because it was a little robot throwing up. But it really speaks to if you put high quality data into a model, into a training pile, you can expect that the function of that model will be as high as it possibly can be. And if dependable models are really built using quality data, for you, Jason, I have a question. Can you speak to what is good data stewardship in the image field, in the imaging field, in terms of curating these data sets, in terms of maintaining them, so that you can eventually have confidence that the image data that we have collected in a database for the past 2 years is going to be able to call correctly all of the things that it sees in an image?
Well, so the first part of the question is what is good stewardship?
Yes.
And so that good stewardship is represented here at the table in many forms. Paul's work that I've done, work at BioHub, where you have data coming in where you have currently largely teams of people, and these are human scientists with an enormous amount of expertise who are working through the curation process to put those data in as solid and as trustable format as possible. I want to come back to as possible. Okay. You saw that. The problem is, and here's it's worth learning from history. So we now have genomics data sets, which we all trust. Those genomics data sets took many years to achieve the level that they now do. In the early days, there were cases where bacterial DNA was stated to be human genome DNA, and there's technical reasons for that. Those data were made public. They were stated to be human genome, and then third-party, the public analysis of that data revealed that there was a mistake. There's similar examples in structural biology, so we now all look at AlphaFold and we're proudly proclaimed that the protein data bank was the source of that. In the early days, there was a lot of data sets in those in the PDB, which had problems.
Okay.
And the way we move forward is we do our best, we curate, we publish, we analyze, we review. And so it's probably true that the data sets we're publishing now in 5 years, well, let's just say they'll be improved upon.
Okay.
Yeah, and in genomics, we've come so far that we even trust now
Right.
that we do and accept that we do have Neanderthal genes
Right.
in our genome.
Right.
Yeah.
Yeah, but most of our work just assumes that those data sets can be trusted and we don't question them, right? But that took 20 years.
20 years.
There's another element in this that's special for imaging data, or at least particular in some ways. You use the word see. Actually, seeing is an active process.
Right.
We don't see passively. When we look at an image, we bring to it some way of focusing and applying measures of salience onto the features that we see in the visual space. And one of the most important things about image data in AI is that the models have to be, this is part of the training, but it's also part of the mechanism of training. It's about identifying that the feature selection that will highlight those properties of the images that will be most salient to the question at hand. And that sort of brings up the main aspect of image data is that it is unbelievably rich. It is so much, when we look at genetic data, each base pair provides one point of information and not more. But when we look at image data, even one pixel contains contrast, edges, intensities,
Spatial
and so on.
information, yeah.
Spatial data. It's incredibly rich, and that's what makes imaging data so special, other than its meaningfulness to us, because we're a visual organism.
Yeah.
And add dynamics to everything that Neil just
Yes.
mentioned, so we can look in time, right?
Through time.
Yeah.
Yeah. Yeah, I mean, it's quite interesting. Only within the past couple of years has the explainability of these models become standard. I remember when we would be reviewing papers and people would say, "Oh, the model thinks that the model says this." And you say, "Why?" And they're like, "Well, we don't know because it's a black box. We don't know." But now, with a lot of the natural language processing, we're able to actually query these models and say, "Can you show me where on the image you're making these decisions from?" Has that been kind of like a insightful kind of moment for you as scientists to say, "Oh, we weren't even looking at that"? Or the attention that these models are giving to different aspects of images is something that a human, because that's really where a model becomes powerful, is that it is compensating for what we as humans are limited in doing.
I think what's happening in those examples is that you are seeing things that, first of all, you expect, or at least that you're able to validate.
Hope, right?
Yeah, exactly. but then there's new surprises as well. So we're looking at a biological mechanism. Okay, it's occurring in this part of the cell. That makes sense to us. But look at this other thing that is highlighted as attention, and that's where the surprises are. That's where the discovery is.
There's also I think one of the walls that we do need to break down. I'll anticipate the last question. Is that we need to break down. we need to break down the distance between the people who make the models and the people who use the outputs of the models, because a critical step in making a model is how one is going to take that image and encode the data in it, encode the features in it in a way that then is used in an AI model. And that encoding step is fundamentally determining what kinds of features are highlighted by the model and used to define the differences that we're looking for. And curiously, we've to date largely ignored the importance of that, and users have used AI models relatively blindly because the explainability criteria that are used are very broad. They're very fluffy. Yeah. You still need to know. We want people going forward to really know how that output was generated, what features were used, and why.
And I think that will be critical if we actually want to start to understand how to intervene within the system.
Yeah.
How do you think about therapeutics in a directed way? So we have to bring down from this black box understanding. We might be able to predict what the outcome of the system, how it will unfold, but we need to understand what are the control knobs, what are the mechanisms that we could help steer the system into optimal functioning. And I think imaging and those large language models are now giving us the ability to start to understand this. So it's taking it from a black box into a mechanistic.
Which is great. I think like something that I have noticed in a lot of the conversations these past 2 days, I don't know if anyone who's been to any other AI talks, is that the human element really gets lost when we talk about AI. Like we talk about AI doing all of these things from end to end, and being left with this question as like, well, what about us? What about who is it serving? It's supposed to be serving us. We're supposed to be participating. And something that we find quite interesting at Nature in the review of a lot of AI models is that we find that in the testing of these models, when you put a human against a computer, sometimes depending on the task, the computer does a great job or the human does. But when you put them together, they always outperform each other individually. And it means that humans will always have kind of a connection to AI. But Martin, as like the head of an institute, I did want to ask you a question about how we train individuals and how we're training kind of the next generation of scientists to develop AI, but just scientists in general who will live in a world where they will be breathing AI with every single experiment that they do. And how do you prevent de-skilling? So AI just replacing people being trained on a certain way, but also how do you keep trainees kind of moored to the foundational biological and technical principles while still encouraging like a fluency in data science that it seems like everyone needs in order to interact with these models now?
This is such a great question. And when you told me that the question actually, this would be my first question. I mean, this is perfect because even before I knew you would ask that question, I actually wanted to make a case, regardless of what you ask, a case about human intelligence, really. Okay. Because I think we can always confuse. I mean, we just discussed, and I think that's absolutely true, AI, especially in imaging, is accelerating, removing major bottlenecks that we had in the past. But what AI cannot do, now at least, and who knows when it's going to be, is actually really deciding, okay, what's the next question that I'm going to, what are really creating this explanatory knowledge? That's a human domain. So I think it's not just serving us, it really needs us as humans. And that also makes it very dangerous. I think you mentioned genomics data, which is not easily accessible to us. And I still remember a colleague of mine, actually a genome person, when I showed him images of protein-protein interaction. It was just at the time when you could do proximity ligation. You could actually literally show in a cell if two proteins get closer than a few angstroms in vicinity, right? Amazing. And he thought, well, seeing is believing, because he didn't believe it at the point based on biochemical data. And obviously it's a bit ironic, seeing is believing now in times of fake. So I keep thinking of that actually. No, he might rephrase it anyway. But it really, I think, shows that we have, unlike proteomics, data genomics, with images, we have a much more intuitive approach. So we actually believe images, and that's also dangerous. And we talk a lot about hallucinations in AI. Well, we had hallucinations in imaging a long time, we just called them artifacts, right? So imaging has always evolved also to managing artifacts. And I know I could many stories. But that, I think, is particularly relevant in this question, that if you have students and postdocs and trainees, people work with it, how do you actually know now and pay attention to, okay, this could be an artifact, this could be real. It is obviously prior knowledge that also you don't carry necessarily with you in AI models. You have to bring this sometimes even with human intuition. The way we deal with this, and I think it's absolutely, you need literacy in multiple fields, and I think you just said this, right? You need to actually bring this closer together. The way we try to do this, we have in our graduate school a system where students are encouraged to rotate between different fields in computer science, in the life sciences, and then really foster a highly collaborative culture that allows students to really interact and ideally be embedded in different labs. And I think we need this not only in the life sciences, but computer science with physics, chemistry. So it's transforming all different areas in science. And I think this is really essential for people to navigate this, to actually also develop better models in the future. And I think this is something that is really upon institutions to take very seriously. And I think one of the problems that many big universities have is this siloed department structure, which is not helpful. And it's why we try to build an environment where no departmental structures and the barrier to actually start to discuss and learn across different domains is really part of our DNA. And I think it's essential.
I mean, one of the things imaging is it's a quantitative science, and it lends itself to high-end analysis. And sort of following what Martin is saying, there has been siloing. So people who were, were experts in high-end image analysts were a certain type of person, and you would be lucky if you had someone like that in your lab. Well, guess what? All of that capability, I won't say knowledge, but capability now starts to appear in Cursor, in ChatGPT. So people are now writing code, so people who have less formal training can rapidly get that experience. And for me, I think the most exciting thing is it's that speed. They can access something and a capability so quickly, test it, refine it, and that, in just a few minutes. Whereas before, we might have said, well, yeah, actually, there's a good graduate course you can go take.
Right.
We'll see you in 6 months, right?
Yeah, we're finding it accelerates the pace of every scientist. It's making every scientist a better scientist when they can adopt both AI tools and imaging tools. And then our ultimate goal is to really reduce that space between bench and bedside. So we can start asking more complex questions. We can start understanding how therapies affect the entire biological system. So it's incredible.
So this is really like, having data hot off the press, as my old PI used to say, where you're like, we can see this protein-protein interaction because, you said that you're dealing with, subcellular, and really saying, okay, let's take it immediately to how does this help us better understand disease and treatment?
Yes.
Yeah.
I guess two things to just add to that. I mean, I think now when we talk about seeing with imaging, we're not necessarily talking about qualitative seeing, we're talking about quantitative seeing. And that actually adds a set of dimensions to it that greatly enrich our ability to make inferences from it. But the other point I was going to make is we hear a lot about, well, in the discussions today going between sessions, there's been a lot of discussion of bias. And coming as a clinician, I think the bias, I just want to introduce, the other thought that, in fact, we live in a world of biases. So a radiologist interpreting images in a typical way brings the bias of the last images that they saw and the particular training sets that they used. If we look across radiologists, and I've done this in the context of brain images for multiple sclerosis, if we ask them to count the lesions in a brain of someone who has multiple sclerosis, the image counts, just the counts of bright spots in the images, vary dramatically between radiologists. And it's not because they can't count or because they're not spending enough time. It's because their bias towards what constitutes a pathological feature is idiosyncratic. And I think what AI is beginning to do is it's beginning to teach us that we can bring consensus reads to images. And although those can still be modified, of course, on an individual level for a clinical decision, those consensus reads introduce a degree of harmonization that I think promises to advance medicine as well as its applications in image science.
Yeah, I think that, it's dealing with kind of like we need to find a consensus. I think that's absolutely true. There always is a question when we're talking about bias as to who gets to make the decision about like what is the consensus, and are they biased in themselves, you know? And I think it's something that we've heard a lot about in the past couple of days, about bias sometimes being baked into models, and how do can we as researchers really do our best to make sure that we are not incorporating our human biases into the models, especially, like you said, for some of these models, you know, the structured data is from someone who has labeled which are the spots, you know?
George, I'll be very brief to let my colleagues speak, but you know, I deal a lot with very large epidemiological data sets. There's always bias. The challenge is to recognize what the biases are. You can't remove bias from any data set, but what is important is to recognize that bias. And I think that the discipline around generation of AI models and identifying the provenance of the data is introducing everyone to the notion of being explicit about identification of biases.
Okay, now that's quite interesting. Yeah.
Following on from Paul, we accept human bias because, you know, so radiologists are human. Okay, that's part of the story. We now have an opportunity to balance, if you will, through the incorporation, you know, the explicit choice of what data sets we're working on, what cell types we work on, where are those cells sourced from, what types of individuals are they sourced from. And so I think for the first time, we now, you know, rather than dump petabytes of data on a poor radiologist and say integrate this into your understanding of the scope of what you need to see. Now we have a way of informing the models. That we have a lot of work to do. This is not going to happen in the next couple of weeks. Okay? but there's a way forward, right?
Do you think that with the adoption of AI, and now everyone just being so cognizant of how data manifests itself and is used by these models, do you feel like in your teams you've become more conscious of where is this data from? Who is this data set? Is it balanced? Is it sex balanced? Is it ethnic balance? Is it age balance? Like, are we really trying our best to make sure that we are not incorporating just kind of skewed data? Because, you know, you could say, let's use the UK Biobank, but of course it's the UK, and so it's restricted to the demographics of the UK. I think that, maybe 10 years ago, you would just say, that's just people, the UK. Now we are much more cognizant of the fact that that is, that is a subset of humanity. You know, it's not representative of the rest of the world necessarily. Not to say that we can't learn things from it, but, but in your teams, do you find that like there's more scrutiny and more care given for each step of the way?
Well, absolutely. I think we talk a lot about biases, rightfully so, but also we just talked about provenance. I mean, the quality of the data will improve dramatically, and not only just analytical data, also the pre-analytical data. I mean, how do we actually prepare the tissue? What is it coming? Not just, sex, and like, where is it come? How was it prepared? That can all be automated and should be automated. That's a huge source of the problem that we can't reproduce data also, right? So the pre-analytical part, then the analytical part, and then the sharing part, that's open sharing of data, that data will be reused. So that's typically in the past, we obtained imaging data, curated, published it, dumped it in a database, and didn't expect it that anyone else, maybe other than a few people, will actually scrutinize it. Well, that's going to change dramatically, and so the quality of the data, really, the whole provenance, I think, will lead to that. Yeah, so I think that's a huge improvement.
How about you, Stephanie? Your team?
Yeah, I think overall we've been giving this quite a bit of care over the years and really thinking about diversification of the data that we're obtaining and pay quite a bit of attention to this. And sometimes it is, so you want the models to be able to be generalizable, but sometimes it is those features or those uniqueness about sex or other things like this that will be really important to understand how the system is operating. And so we overall pay quite a bit of attention to this, and it's only increasing from here on out.
Good.
And this diversification of data sets is really critical. It's diversification and then maximizing scale. Because hitherto we've tended to just grab any data that sort of generally fit the bill and mushed it together and derived models from that, because that was the only way we could get the scale of data necessary to train the models. Yeah. What we need to look forward to is an exciting future in which we have large scales of diverse data, and then we can actually go back and build models from individual data subsets that then will tell us, for example, how to deal with health challenges in women, specifically because the models are developed on women or men or Africans or Northern Europeans, so on.
Yeah.
And ultimately, we want to be able to get to a understanding of how to, for an individual, how are we going to interact? What's unique about you, and how, if you're in a disease state, what is it about your disease manifestation? So ultimately, what you want to get down to is the individual level.
Individual level, yeah.
No, I was just wanted to add, because that will also fully utilize the federated approach model, and so there's also a lot of innovation coming from the AI side that's necessary, and I think will be the future, that we have enormously diverse data, much higher quality, and so this is really good news for all areas of science.
Yeah. I want to take a moment now to open to the floor for questions. So if you have a question, there should be some of our helpers with a microphone. I think we have some individuals over here and someone in the back.
Hello,
Hi.
Karit Marte from the Medical University of Innsbruck. It has been mentioned that AI can come in where it is hard to fill in gaps that are hard to reach experimentally. I would say a huge opportunity there by looking at fusion of various imaging modalities. There have been efforts towards a correlative microscopy, combining electron microscope and fluorescence. But I don't actually see a lot of things emerging on the AI side there. I was wondering, could you comment on that, whether people do this more and more to have this fusion?
I think this is happening more and more. So one example I can give is with the ERSF facility in France. It's using a technique to be able to image whole human organs down to cellular resolution.
Oh, I didn't know this.
And so they use high energy X-rays to be able to do this. And then they're using AI techniques to be able to, so now they get this really high structural map. It's post-mortem human tissue. But now they're also imaging those same organs with device like CT scans, so you're able to actually start to see what this would look like in a live human, and then start to make these correlations. Now, features that look like little blobs or smears within what physicians were seeing. Now we understand what that structure, that inherent structure is, and AI techniques are helping to facilitate this.
Yeah. But can I just add also that you can achieve now also in part of a combination of new methods to prepare, like also pre-analytical, 20 nanometer, like EM-like resolution on live tissue samples, like brain slices, for instance, right? And get transcriptome data, proteomics data on top of it, really. And I think, and so I think that's where also, that's even beyond that, yeah.
Yeah. We can keep going.
Yeah.
Yes.
Did you have a question?
Emphatic yes is the answer.
Hello, my name is Maria Leudold-Krüger from Pico Quanti in Berlin. Thanks for the insightful discussion. It's really exciting. So we all know, so AI can help us to visualize and can help us to quantify and to recognize. But at least in my mind, there's still a gap between recognizing, quantifying, and actually understanding, as in mechanistic understanding. Which you need if you want to cure diseases, for example, and also to close the loop based on your understanding, you develop your new hypothesis, which, at least for fluorescence, informs what you actually label. So what you actually can even see in your new experiment. So if you could comment how AI might help this, because I think even if you have petabyte of data, that doesn't necessarily lead to better mechanistic understanding.
So like knowledge versus understanding. Yeah.
Yeah, so I think it's a great question, and I think you're talking about one of the real frontier areas right now, and that's really, Stephanie referred to it right at the beginning, and that's really trying to capture dynamics, because what we're trying to do with mechanism typically is to predict what is going to happen next. If we know this, if we know a state now, what's going to be the next state? And AI is becoming a really powerful tool for knitting together images that are acquired in different states to create most probable trajectories of evolution. And that's just another way of stating that you have a causal chain. And I think that if we add to that, the increasing focus on adding dynamics into all of our imaging techniques, I think it's going to really completely change the game, and we'll be thinking about causal modeling in a completely new way.
And what is starting to happen is that the concept that Paul is talking about, but applied to an oncology case. So what I see is a pathology slide, and I'm able to profile and make a statement about the current diagnosis, but also the disease trajectory with high certainty.
You did not like that answer. That answer is done. Thank you. Yes, we have someone in front. We can get you a microphone. Yeah.
Thank you. My question was about, maybe related to the previous one a little bit, and about feature extraction. We had engineering feature previously before the AI boom. And I would like to hear your comments about opening the AI box and looking from the learned features. Where is the place of AI in that? And also another small question about 3D rendering. We had a while ago algorithms like NERF, and now we have Gaussian splatting. So what is the place of such new rendering methods compared to the classical ray tracing or matrix multiplication methods? Thank you.
Okay, features. It's sad to say a lot of the features that people have been using, those concepts are a couple decades old. They were established from the computer vision community way, way, way back. These are the people that are looking at bicycles on roads and things like that, and we imported them. They work pretty well. And so what these new methods give us is a whole new way of being able to pull out information out of these images, these late space representations. And there's just a raft of papers showing up in many journals where people are trying to experiment with this. So it's going to be an incredibly exciting few years as we work through this, right? And it gives us capabilities we've never had before. Visualization, that's probably your department.
Yes, I won't say anything. I'm not sure I will give you much insight, except to say that I think two things. One is the models that help us understand more deeply what features are being used when we do blinder feature selection and coding. They're still relatively crude, and they're very difficult to apply on an individual data set. They're better to describe trends across very large sets of data sets. So my sense is that they are less useful at this point, but there is a subset of tools that are beginning to overlay statistical-like frameworks on this, and they're actually providing us likelihood estimates on features contributing or being there, and they may give us a new way of thinking about what the meaningfulness of features are. I think it comes down to a fundamental change in our relationship with an image. We don't think about this very often, but what we interpret as something that is real in our visual space is actually based on it's an interpretation that we're making on the fly, and it only has meaning so long as the predictions that for following on in the next moment hold. And that goes down to how we recognize ourselves hallucinations. They don't conform to this sort of model. And I think when we approach AI tools, I'm not talking about hallucinations in AI, but I'm just talking about the basic ways in which they function. We're going to have to apply some of the same kinds of consistency rules to our interpretation of them.
And then how about for 3D?
Yeah.
3D imaging modalities.
Yeah.
Yeah.
No, you go ahead.
Yeah.
Yeah, I think both the sort of this n-dimensional space, that's what we talk about quite a bit. We're just beginning to interrogate this because the techniques to really start to visualize and 3D, 4D to be able to add on the understanding of also gene expression, the transcriptomes, other things about cellular state. We're starting to bring all of this data together. And so how we do feature selection is a emerging field right now.
I mean, the only thing I would like to add is also, especially in the across different time scales and different modalities, I think it's an exciting time for mathematics because there's still even some basic fundamental mathematical models we don't have yet, right? Okay. So I think it's not just computer science, AI, also mathematics is continuing to be extremely interesting for the future related to those questions.
And just to go back to your question specifically, I think what Stephanie's highlighted is that I think now we're not thinking about how do you do 3D rendering better, but the question is how do you do ND rendering in the best way possible.
Yeah. Did we have any more? I think we have time for probably like one or maybe two questions. Right here in front, they'll have a microphone for you.
Hello, I'm Bernadette Stolz from the Max Planck Institute of Biochemistry in Munich. I was wondering, so you mentioned that AI models are only as good as the data that you put in them, and we need really high quality data for this. But the issue is often the highest quality data comes in very small quantities. So how can we really use AI to make the most out of small data sets? Or is it just a question of waiting for technology to improve experiments to become less costly and then applying AI?
I'll start. I think there are two answers. The first is that if the value of the data is high enough, we can learn from small data sets how to create big data sets. And a really good example of that is 10 years ago when I was first approached to think about the UK Biobank clinical imaging data set, the leader of UK Biobank said, what we'd like to do tens of thousands of fMRI scans. And I said, that's absolutely impossible. Absolutely impossible. These are hard studies. We typically are lucky to do 25 good ones. And I'll tell you that just a few months ago, we hit 100,000 images in UK Biobank of brain and other body parts. So it can happen. But I think the other answer to the question is we haven't mentioned it, but the key to imaging data or any other thing that we use for AI is having the metadata that accompanies it. So if we have small data sets that are extremely well annotated, we know how the experiment was done, what the conditions of the experiment, et cetera, then actually that contributes to creating a better model. And so they can be aggregated as small data sets more effectively, more informatively, because we're carrying with them not just the image data, but the provenance and methods associated with it, which all can be encoded at the same time.
You're right. We have to accelerate this. So as Paul said, if we look back 10 years, so Paul mentioned the medical imaging space, the biological imaging space, we started the first public data sets. There's now over a couple petabytes of data publicly available, and there's more coming. The model that we've been using, and this is similar to UK Biobank, is that we will collect data sets and then we will concentrate them in a single location-ish. And we're following models that were established in genomics and structural biology. But to achieve what you're referring to, we are going to have to build a federated system where now multiple efforts, institutes, programs are building up these data sets. They won't all transfer their data to a single location. It's just too big. We'll need to have that federated system, and you need to be able to identify the data because it has that common metadata capability, so it's interoperable. But then we need to be able to find that data. So you need to be able to have a chat GPT query or whatever, find me data that looks like whatever, and that needs to work at a level that is meaningful for science. And that's the next step.
Did you have something to add, Stephanie?
Yeah, so at BioHub right now, we are focused on collecting vast data sets and making sure that all of those data sets are publicly available and training the models on those data sets and also making sure those models are publicly available, which allows us to start to test the generalizability of the model on some of the smaller data sets. But I do think structured data is important, and some of these large-scale data efforts are required at this stage to be able to drive the field forward.
And I think the only thing I can add from the perspective of an institution, I think the federated system is one way to go that all institutions have to go. But also within an institution, you can, by centralizing imaging, actually having, instead of everyone has his own imaging modality, centralize it, have people who are AI trained, centralize, and then add additional modalities, like even from genomics, and to really create smaller but highly annotated, high-quality data sets, and then load those up in a federated system. And I think that will really also accelerate real discoveries.
Yeah. Okay, so now it's time for the falling walls question. So I think, probably we have 3 minutes left. I guess a question is, what in a short would you see as the future of imaging, and if AI plays a role in that, and how? And then what needs to break down? What kind of wall do we need to overcome to get there? Do you want to start, Stephanie?
Sure. Right now, there are a number of different modalities or fields that are focused on different levels of investigation. And so there's folks that focus on electron microscopy and then studying individual proteins, and then groups that are focused on light microscopy approaches, studying the dynamics of cells. I think what we need to be able to do is to be able to build a integrative map of how the system is functioning. And I think that when you do that, what you recognize is that there's emergent properties that emerge. And that it's really important for us to be able to understand those emergent properties, and this requires us to break down the walls between disciplines.
Martin?
Just because we haven't used that concept, also AI helps to democratize imaging, right? There are still many examples, and we talked about some, where only a few places could even do this imaging. Now you can do things that could only do with extremely expensive, highly accessible, hardly accessible instruments on machines that many institutions have around the world, and that's another level that is also really unleashed by AI, and I think that's a fall that is crumbling or disappearing.
I completely agree. Take those and add this federated capability, so that there's not a million sites worldwide, but there's probably a few tens that are taking the kinds of data that Stephanie and Martin just described, making those data available as reference data sets, models training on those. That will change the world.
Yeah, and I cheated a bit earlier in breaking down a small section of the wall, but I think the other thing
chipped away.
my big boulder will come to the wall. I think the next big frontier is to bring the AI to the way we acquire the data.
Yeah.
So every imaging technique is benefiting from the pilots that have shown how AI can transform the quality, the speed, and therefore the scale with which the imaging can be generated, even down to the point of sample preparation for complex problems like cryo-electron tomography.
I guess because then we get the high quality data.
We get high quality, harmonized data at scale.
And a rapid feedback loop, which is really important, because now you can build the models, validate, and test them in a rapid cycle.
Yeah.
All right, everybody.
Great.
21 seconds
Final
left.
words?
Final words are...
Our moderator.
I mean, I want to say that it's been really incredible. I feel like we've touched on so many different aspects. I think that imaging is such a special aspect when talking about AI because it is tactile, because we do see, and because really it lends itself so well to all of the strengths of AI. I think the genome AI, probably that field is in a little bit of existential crisis always, but the imaging field really does seem to have like a North Star in terms of where they want to go. And the progress has been swift, and it's been really incredible to watch it from the editorial side and to work with scientists who are every day kind of breaking down walls. So with that, I want to close the final plenary table discussion of the day. I do want to invite all of you to join us for drinks. Everyone loves drinks at the evening reception. Or if you don't want to have drinks, there's also the Berlin Science Week Forum. I want to thank all of you again for joining us at Falling Walls 2025. This is the last plenary table of the day. I want to thank all of our partners and supporters, and I look forward to seeing all of you tomorrow or next year. Yeah.